Hi, dear students. As a part of your micropropagation technologies, so you will be learning in this particular lecture on various tissue culture methodologies and also how to follow the sterile techniques. In the previous classes, you have already studied the very basic things like how to maintain the aseptic culture and why should we go for maintaining and how it is directly related to achieve successful tissue culture plantlets production. And now we will see so what are the possible ways of contamination and where we have to actually concentrate on avoiding the contamination and other things in order to achieve the success in tissue culture. We'll see one by one. So normally the basal media will be having these are the things like macronutrients, micronutrients, vitamins, growth regulators, sugars as well as gelling agent and also water, that is distilled water. And you already know what is the role of these macronutrients or micronutrients, like how the plant outside it grows, like with the when we are putting some plant in soil, the soil naturally will be having macronutrients as well as micronutrients, so that will nourish the plant. But it is a whatever tissue culture techniques or whatever the concept tissue culture, here we have to provide the same condition for the plant to develop properly and also to produce a healthy planting material as well. So whatever vitamins, amino acids, everything we should supply artificially, while preparation of these macronutrients, micronutrients, stock solutions are the totally entire media. So there will be a contamination. So this has to be avoided in order to achieve success in tissue culture. Normally sugars. So what is the role of these sugars? We will see. So it will avoid the bacteria. Suppose if a excess sugar is applied to the particular media, tissue culture media, it leads to the like occurrence of some infestation or like contamination, we can say. Then suppose if we are using some of the things like other things like gelling agent in order to get proper consistency and other things. So we will see next how to establish this aseptic culture in order to achieve contamination free tissue culture plantlets. So these are the various equipments we can use, like starting from the media and whatever the petri plates we are using, it should be sterilized properly, and even whatever the other equipments and along with some forceps, even charcoal and other things, it should be properly used or it should be sterilized before usage. And other equipments like autoclaving or like how to do the wet sterilization and also dry sterilization. Normally autoclaving we will do at so for about 15 to 20 minutes at a temperature of 121 degrees Celsius with a pressure of 15 Pascal. So it may be like vertical or horizontal autoclave based on the availability. So this can be used as a like a part of wet sterilization. Sterilization, that is wet sterilization. Here, the whatever the moist heat it developed in that particular autoclave, that helps in killing the microorganisms. Even here we can sterilize the glass vials, whatever we are using, all the glass vials we can sterilize. Even petri plates, before usage or before start of any preparation of media we can sterilize. Even after preparation of the media also, the media can be sterilized here. So that is the one thing. And dry heat sterilization, I think all of you have seen this. So it is a hot air oven, we can use this for dry heat sterilization. Like it depends on the material which you are using. So same here, like autoclave here, instead of moist heat, here dry heat will be used to kill the harmful microorganisms. like same way, like petri plates and other the vials we can sterilize here. Then we can use it for following some other tissue culture steps. And filtration, radiation, these are the other things we will see one by one. So normally, what are the possible ways of contamination, or how the tissue culture media get, our tissue culture techniques will get contaminated? There are different ways here, like culture vessels, whatever the vials or vessels we are using for preparation of the media, that is the one important or the very basic possible way of contaminant. And medium, whatever we are preparing, even there are different steps, like we can prepare directly the ready-made media, or we can separately prepare macronutrients, micronutrients, growth regulators separately. So in this process there will be a contamination. And whenever we are selecting the explant also, the explant should be always, it should be selected in a contamination free zone. Suppose if it is not sterilized before usage, again it leads to the contamination. And also whenever we are transferring that particular explant from the after selection, whenever we are transferring it to the media, so in that time also we can experience the problem of contamination. And whatever the equipments you have seen, forceps or glass vials or any other things, also leads to the contamination. And whatever the growing, like after all these steps, again finally we will be keeping this um, uh, media as well as media containing explant in a culture room. So there also we can expect the contamination. So we will see, like suppose if it is, the contamination is through glass vials or medium, so this can be avoided by autoclaving process. Suppose if it is through explants, so we can use some sterilants, like surface sterilants, like sodium hypochlorite. Normally they will use this, or ethanol we can use. Suppose if it is environmental control, or like um, the infection is occurring through the particular environment, then we can use laminar airflow. Suppose if it is any equipments or instruments, say for example, if we are using forceps or glass vials, then we can go for sterilization. It may be wet sterilization, or it may be dry sterilization. And whatever the cultural room, that in that we can maintain proper light, temperature, and it should be always dust free. And the person who is entering into that particular growing chamber, he or she should have the proper lab coat and other things, like all the tissue culture laboratory safety practices, along with the aseptic practices the person should follow. Then only we can avoid the contamination. Then we'll see like how to prepare this growth regulators, or what are the growth regulators, and how the contamination will occur during the growth regulator preparation. Normally we will use different types of growth regulators, like gibberellin, abscisic acid we will use, and also urea, certain vitamins. So this can be avoided by autoclaving, like the contamination can be avoided by autoclaving. And the next is like whenever we are using some filters also, by using the filters also we can avoid the contamination. Then the filter size or pore size should be 0.2 to 0.45 micromolar. So it will helps in avoiding the pathogen. And even we can also go for autoclaving, and then we can also cool these things to avoid the contamination. Suppose if there is a excess contamination in explant, again like whatever the presence of microorganisms on the particular surface of leaf or bud or anther, embryo, or irrespective of the explant, normally they will recommend sodium or calcium hypochloride as a disinfectants. So sodium hypochloride 0.5 to 1% is recommended as a disinfectant. And even we can also use if we, this one we can also use whenever the tissues are damaged. Even ethanol can be also used as a sterilant. Laminar airflow normally here the I have already told we can use autoclave or laminar airflow and even filters. Normally here we can see the HEPA filters like high efficiency throughput phenotyping these HEPA filters will helps in avoiding the very small minute microorganisms which we cannot be controlled even in autoclaving or by using the or sterilizing the things in hot air oven. So this can be completely avoided by because whatever the high speed air which is transferring, that will helps in avoiding the problem of entry of microorganisms into that particular laminar airflow. This contamination may occur mainly whenever we are transferring the tissue culture plant like explant into the media. During that time we can expect this problem. Normally for this, even before you entering into the laminar airflow or carrying any work under laminar airflow, we will always recommend to disinfect our even hands and also the surface area. So that is ethanol is commonly used sterilant here. And if the contamination is more in culture room, how to avoid this? Normally, we should always keep this culture room in a good condition without any infestation and all. And even whatever the hair entry and other things we can restrict. And as I mentioned, they can use some they can use lab coat, and they can also practice laboratory safety practices in a proper manner in order to avoid the contamination or in order to avoid the any entry of harmful microorganisms. So, in this particular lecture, you have seen the many things which were which are the possible ways of contamination in the tissue culture process and how to avoid this, and what is sterilization. Sterilization is a aseptic condition or maintaining aseptic condition or killing the harmful microorganisms. This was already explained in the very basic class. So sterilization and in the two types what are the equipments used and how to maintain this particular equipment. So all these things you have seen. This is very important topic like sterilization and how to maintain aseptic condition and what are the sterilization techniques and which are the possible ways of contamination and how to avoid this. Thank you.